Virology Chapter 3 Online Flashcard. So, question number 1. Gene regulation process whereby certain exons are included or excluded from mRNA, resulting in a single gene that codes for multiple proteins. For example, the viral mRNAs representing segments 7 and 8 of influenza A viruses undergo this. Alternative splicing. 2. Procedures designed to prevent contamination from organisms involves using the strictest rules based on what is known about infection prevention in order to minimize the risk of infection. Aseptic technique. 3. A virus that infects and replicates within bacterial cells. Bacteriophage. 4. Systematic search for new therapies to treat diseases based on biological resources, mainly plants, gaining medicinal plant knowledge from traditional healers and farmers in diverse cultures for commercial valuable. Bioprospecting. 5. The process by which the cellular ribosomes recognize the 5' M7GPPP cap present on the 5' end of messenger RNAs, all eukaryotic hosts and some viral, in order to direct translation or synthesis of proteins. Cap-dependent translation. 6. The process by which the cellular ribosome bypasses the 5' M7GPPP cap recognition requirement in order to direct the translation of viral messenger RNAs. Cap-independent translation. 7. A family of proteases that play a role in apoptosis. Caspases. 8. A large fibrous protein that forms a basket-like network around vesicles. Clathrin. 9. Specialized regions within the eukaryotic cellular plasma membrane that appear as invaginations that are coated on the cytoplasmic side of the membrane with dark material, may be involved in viral entry. Clathrin-coated pits. 10. Transport vesicles coated with a lattice-like network of this protein. Clathrin-coated vesicles. 11. A second cellular receptor necessary for viral entry into a host cell. Co-receptors. 12. The part of a eukaryotic cell in which processed messenger RNAs exit the nucleus and are translated. Cytoplasm. 13. Three-dimensional structure composed of protein filaments that helps eukaryotic cells maintain their shape and internal organization. Cytoskeleton. 14. An infectious hepatitis B virus particle measuring 42 nanometers in diameter that is found within the body of a person infected with hepatitis B. It contains a double protein shell, hepatitis surface B antigen on the surface, HBsAg, on the particle, and hepatitis B core antigen, HBcAg, inside the particle. Dane particle. 15. An enzyme that catalyzes the synthesis of DNA. DNA polymerases. 16. A protein that is responsible for endocytosis in eukaryotic cells. Dynamin. 17. Steps involved in the initiation of endocytosis, formation of endosomes, and their ultimate degradation of the endosome and its contents. Endocytic pathway. 18. A vesicle formed by the invagination and pinching off of the cell membrane during endocytosis. Viruses exploit endocytic pathways for infection. Endosome. 19. DNA elements outside of the promoter region of eukaryotic chromosomes that stimulate the frequency of transcription of genes by RNA polymerase II. Enhancers. 20. A human or animal virus that contains a membrane that was stolen from the host cell that is wrapped around the capsid of the virus particle. Enveloped virus. 21. A circularized viral genome that replicates autonomously within the cytoplasm or nucleus of eukaryotic host cells. The provirus of hepatitis B virus is an example of this that is not integrated into the chromosome of its host during replication. Episome. 22. An enzyme that removes nucleotides one by one from the end of RNA or DNA. Exonuclease. 23. Inflammation of the stomach and intestines, often due to infections caused by bacteria or viruses. Symptoms include nausea, vomiting, and diarrhea. Gastroenteritis. 24. A segment of a DNA molecule that provides the biochemical information for a functional product. Gene. 25. Sperm and egg cells. Germline cells. 26. The enzyme that separates double-stranded DNA into single strands for replication. Helicase. 27. The variety of species and types of cells that a disease-causing microorganism or virus can infect and replicate in. Host range. 28. Remnants, DNA sequences present in the human genome that indicate the occurrence of past retroviral infections. Human endogenous retrovirus. 29. Enzyme that facilitates the integration of a retroviral DNA into cellular DNA. Retroviruses include human immunodeficiency virus, HIV, possess this. Integrase activity. 30. A stretch of RNA that is removed from pre-RNAs by a splicing event before the mature messenger RNA can be translated by ribosomes into protein. Intron. 31. A sequence of eukaryotic messenger RNAs that plays a role in the initiation of the translation process. Kozak consensus sequence. 32. A strategy that allows multiple viral proteins to be synthesized from a single messenger RNA. Cellular ribosomes may inefficiently initiate translation at the first AUG present in a messenger RNA, but more often the 40S ribosome will bypass the first AUG and initiate translation farther downstream of the messenger RNA at an AUG in a better context. Leaky scanning. 33. a microscopy method developed in 2006 in which viral components and relevant cellular structures are labeled with fluorescent probes to track the fate of individual virus particles or viral components inside of live host cells in real time. Live cell imaging. 34. The rupture of a cell and the loss of its content. Lysis. 35. The stage of the virus replication cycle in which the virus becomes infectious often involves proteases that cleave viral proteins before the virion is completely assembled and released from its host cell. Maturation. 36. A synthetic cell culture media developed in 1959 by Harry Eagle that is still one of the most widely used of all synthetic cell culture media to cultivate and maintain mammalian cells in the research laboratory. Minimal essential medium. 37. A configuration in which the entire viral genome consists of one nucleic acid molecule. Monopartite genome. 38. A situation in which the entire viral genome consists of several nucleic acid molecules packaged into a single virus particle, or each nucleic acid molecule that comprises the entire genome is packaged into separate virus particles, a property of certain plant viruses. The plant must be infected with discrete virus particles representing the entire genome of the virus to cause infection. Multipartite genome. 39. The average number of viruses that infect a single cell, used to study a single replication cycle of a specific virus in order to generate a one-step growth curve. An average of 10 of this is used to ensure that every cell is infected simultaneously so that the viruses are at the same step of replication in order to study them. Multiplicity of infection. 40. A virus without a lipid bilayer membrane envelope around the capsid of the virus particle. Its surface is composed of a protein shell, increasing its stability in the environment. Example, norovirus and papillomaviruses. Naked virus. 41. A glycoprotein that cleaves the sialic acid moieties present on the surface of cells. Neuraminidase. 42. A viral type of this that is composed of linear RNA or DNA. Non-segmented genome. 43. Large protein complexes that span the nuclear envelope that surrounds the eukaryotic cell nucleus, allows specific cellular proteins to be exchanged between the nucleus and cytoplasm because DNA replication and RNA transcription occur inside of the nucleus, allows mature messenger RNAs to be exported from the nucleus into the cytoplasm for translation, also acts as protective barrier to prevent harmful macromolecules, example nucleic acids from viruses, from entering the nucleus. Cellular proteins that move through this must contain a short localization signal sequence for import and nuclear export signal sequences for export. Nuclear pore complex. 44. Resembles a nucleoside such as adenosine or guanosine, used as an antiviral agent to inhibit the genome replication of certain viruses through a chain termination mechanism. Nucleoside analog. 45. The membrane-bound organelle inside of all eukaryotic cells where DNA replication and RNA transcription take place. Nucleus. 46. Short, newly synthesized DNA fragments complementary to the lagging template strand during DNA replication of organisms. The discontinuous short of these are joined together by cellular ligases during DNA replication. Okazaki fragment. 47. Research studies used to examine a single replication cycle of a particular virus by infecting host cells with viruses at a high multiplicity of infection. One-step growth experiments. 48. A stretch of multiple adenosine monophosphates that was added to the 3' end of all eukaryotic messenger RNAs and some viral messenger RNAs through a polyadenylation process after transcription. This may play a role in protecting messenger RNA from degradation, increasing the stability and lifespan of some messenger RNAs in a cell. Poly A tail. 49. A long precursor protein translated from a viral messenger RNA encompassing several genes, which is then cleaved into individual proteins by viral or cellular proteases. Polyprotein. 50. An enzyme involved in DNA replication that catalyzes the synthesis of RNA primers. DNA polymerases can only initiate the synthesis of a DNA strand from an RNA primer that contains a free 3' hydroxyl group base paired to the template DNA it is copying. Primase. 51. Region on DNA to which RNA polymerase binds to initiate transcription of DNA into RNA. Promoter. 52. The function of DNA polymerases that allows the enzyme to check each nucleotide during the synthesis of DNA and correct mismatched nucleotides by reversing the DNA polymerase direction to 3' towards 5', in which it uses its 3' towards 5' exonuclease function to remove the incorrect nucleotide. After the incorrect nucleotide is excised, DNA polymerase reinserts the correct nucleotide, and DNA replication can continue in the 5' towards 3' direction. The extent of this determines its mutation rate. In contrast, RNA polymerases do not possess this capability, resulting in more mutations during transcription of RNA. Proofreading ability. 53. A retroviral genome integrated into the DNA of a host cell. Provirus. 54. A laboratory technique that uses these and electron microscopy to distinguish introns from exons in double-stranded DNA. R-loop mapping. 55. An RNA-dependent RNA polymerase that synthesizes the viral genome using positive single-stranded RNA as a template. Replicase. 56. The process by which genetic material is synthesized by a polymerase. Replication. 57. An enzyme that cleaves DNA at sequence-specific sites, usually palindromes, used in recombinant DNA technology to insert DNA into plasmids or for RFLP analysis. Restriction enzyme. 58. Nasal discharge, also referred to as having a runny nose, symptom of infection from respiratory viruses such as influenza A and B viruses and rhinoviruses. Rhinorrhea. 59. Occurs when the ribosome shifts forward or backward into another reading frame and continues translating the messenger RNA into an alternative protein not directly encoded by the messenger RNA or two or more different isoforms, proteins that differ in their N-terminus that are in the same reading frame. Ribosomal frame shifting. 60. RNA that, together with proteins, makes up the ribosome. Ribosomal RNA. 61. enzyme that transcribes RNA from a DNA template. RNA polymerase. 62. Eukaryotic enzyme that synthesizes messenger RNA and some small nuclear RNAs, snRNA. RNA polymerase 2. 63. An enzyme that catalyzes RNA using an RNA template. RNA-dependent RNA polymerase. 64. A type of polio vaccine developed using live attenuated strains of polioviruses. The vaccine is taken orally. 7. Vaccine. 65. A type of poliovirus vaccine developed using inactivated polioviruses that are injected into the body. Salk vaccine. 66. A collaboration between traditional healers, physicians, and medical staff to manage HIV/AIDS in Tanzania. The goal is to use indigenous knowledge to alleviate suffering from HIV/AIDS. Tanga Project. 67. A eukaryotic promoter sequence present on DNA that defines the initiation of a transcription start site for RNA polymerase. TATA box. 68. DNA structures present at the ends of eukaryotic chromosomes that are not included in DNA replication. They protect the chromosome from fusion to other chromosomes and degradation by nucleases. Telomeres. 69. Essential eukaryotic enzyme expressed during cell division that allows the cell to utilize an alternate metabolic pathway for incorporating thymidine into DNA. This gene is also present in the vaccinia virus genome. Absence of thymidine kinase activity in host cells and disruption of the TK in vaccinia virus render host cells resistant to toxic effects of 5-bromodioxyuridine, BRDU. BRDU is a mutagenic nucleoside that substitutes for thymidine in DNA, acting as a chain terminator, and causes breaks in the chromosomes of cells. Vaccinia recombinant viruses can be generated by using BRDU selection. Thymidine kinase. 70. Describes RNA-dependent RNA polymerases that transcribe negative single-stranded RNA genomes into monocistronic positive single-stranded RNAs or viral messenger RNAs that can be recognized by the host cell ribosomal machinery and translated into proteins. Transcriptase. 71. The biochemical process in which RNA polymerases synthesize RNA based on a DNA template. This occurs in the 5' direction towards 3' in the 5' towards 3' direction in the nucleus of eukaryotic cells. Transcription. 72. Nuclear proteins of eukaryotic cells that form a pre-initiation complex on the promoter region of genes in order to recruit RNA polymerase II to initiate this of RNA from a DNA template, or the expression of genes/messenger RNA synthesis. Transcription factors. 73. Biological process in which cellular or viral messenger RNAs are translated into proteins by host cell ribosomes, translational machinery. This occurs in the cytoplasm of eukaryotic cells. Translation. The continuation of translation beyond a stop codon in messenger RNA by the use of a special tRNA that recognizes UAG and UGA codons as modified amino acids rather than termination codons, resulting in a translated protein with an extended C terminus, a variation of ribosomal frame shifting. This is used as a regulatory strategy by some plant, animal, and bacterial viruses. Translational read-through. 75. A group of fatal neurodegenerative brain diseases in humans and animals caused by prions, including Kuru, Scrapie, bovine spongiform encephalopathy, BSE, and variant Creutzfeldt-Jakob disease. Transmissible spongiform encephalopathies. 76. A segment of DNA that is roughly 100 to 10,000 base pairs in length that moves around the genome by a cut and paste mechanism. Nearly half of the human genome consists of mostly defunct elements of this type. Transposable genetic elements. 77. An enzyme that binds to the end of a transposon and randomly catalyzes its movement to another part of the host genome using a transposition mechanism. These genes are carried by retroviruses known as integrases and bacteriophages. Transposase. 78. The separation of the viral genome from the capsid after the virus gained entry into a eukaryotic host cell. Uncoating. 79. A fully assembled infectious virus particle. Virion. Number 80. Large cytoplasmic compartments in host cells where viral replication and assembly occur can be thought of as a viral factory. Viroplasm.